Video? Oh,
Yes, thank you,
okay.
sir.
Yeah. Uh, yeah. Good evening, all. Um, I am Professor Sudhakar from Department of Biotechnology, Manonmaniam Sundaraj University, Tirunelveli. Uh, I'm so happy to be one among of you, and it's a great arrangement of meeting leadership conference. And the previous speaker nicely presented the sustainable development in Asian countries from Dubai um, and India, and also you know uh, East Asian countries. Such a great effort and a very great ambition for development of uh, people's living standard in this area. So here in India, the situation is much different. The R&D is not appreciated for last 10 years, and not adequate funds are there for researcher. And some, with adequate available fund, we could do serve for student in the area of higher education. I hope in coming days the situation will change. And today I'm going to share some of my experience in the area of regeneration. Actually, for last 18 years, we are using earthworm uh, for our studies. It's a wonderful animal for regeneration studies. We used to cut the earthworm head and tail area, and we are studying cell and molecular biology and also some metabolomic area of studies to understand the regeneration. And so far, we have published around 50 papers in different journals. And there are two kind of output in this effort. One is research articles, another one is nice students. And the students are in different countries now, and they are working in different area of regeneration right now in different parts of the world. The slide I'm showing here is a nice output of our research. During regeneration time, we are harvesting the metabolites from the earthworm. And the metabolites we studied with analytical tools like mass spec, LCMS, and many other the fine instruments. We have listed more than 3,000 metabolites, and based on molecular weight, we have annotated them also, and then we tested the biological property of the metabolites using mouse as a model. So mouse pinna, especially the bulbsy white mouse. If we puncture the ear, the scar will be the permanent. There will be a hole in the mouse pinna till their end of life. So it's a permanent scar will be generated, and the tissue won't grow again. So that's known around the world. People are using uh, the mouse for research purpose in many laboratories around the world. So we use that mouse as a model animal, and we injected the metabolites of regenerating earthworm. Simply, I can explain it slightly. We used to cut the worm at 10th segment, and after third day, the worm starts formation of blastema. And on sixth day, we used to open the worm, and then all the tissues around this regenerative area, we are collecting it. And those tissues, we are uh, um, we are immersed in uh, 70% ethanol. So, and then we are slightly we are grinding it, and the idea here, all the metabolites in the tissue should reach the ethanol. And then after slight grinding, we are uh, we can see the many precipitates, and then we are going for a small spin around 5,000 rpm, like 5 minutes. So all this sediment will be pelleted, and the clear solution will be taken, and then that solution further will be concentrated. And then we are using this solution as reagent, and we are injecting this solution near the amputation sites of the mouse pinna. So first we are puncturing the pinna, then we are loading a small volume of the reagent in the proximity of the wound site, and then we are uh, observing it. So every uh, second day, we are injecting this small volume of the reagent, and then around like 28th day, we could see complete healing of the mouse ear puncture. So such a uh, observation was also made by different scientists by different treatment. For example, the knockdown of P21 gene that triggers a regeneration, that has been reported. And likewise, in this P21 pathway, different genes was taken, and the regeneration studies have been completed. And here we use the metabolites from the metabolites from earthworm, and we have shown the regeneration ability of a mouse phenom. So the extract, the earthworm extract, can trigger regeneration in the mammalian system. That's a point here. And uh, we tested some but cell biology also, and we took cells from the uh, injection site. We could see the cell division of cartilaginous bone, and also sebaceous gland, sweat gland, and other tissues in this mouse phenom. So we think this is a nice reagent, and we can go for further studies, and we can make a medicine that we can call it as a regenerative medicine, and that's our plan. And also this reagent is a good, very good, very good for wound healing also. And we created wound in a different animal model, and then we applied this reagent, and we could see really faster healing of wounds. Likewise, we are progressing the experiment, and to design a molecule, to design a formula which can help people to recover from the wound, or and amputated tissues or organ. And that's our goal, and that's our we are trying for it. And I could say one thing, and for us, wound healing is a long term. For the earthworm, it's really a simple matter. For example, if we cut the worm can heal the wound uh, within 1 hour. We have data. So it's such a fast wound healing power the worm has. So it has very different biology, and during that time, very nice molecules are produced. Those molecules can be used and can be utilized for better treatment for for human healthcare. That's for our wish, and we have some data in this area, as I shown here, and I hope we can come with a product to help people. Yeah, I wish this organizer of the program, Dr. Ram, and of all other organizer of this program. I wish all of you for the grand success of the conference, and and also I thank the coordinators for inviting me to present my um, thoughts in this prestigious conference. Yeah, thank you so much. Yeah, thank you so much.
Thank you very much, sir. Thank you. It was really a very unique thing that you have presented to us, and it is a great research. We wish you good luck, and I hope that it is going to be beneficial for others. And over to you, Dr. Ram.
Yeah, thank you.
Really, thanks for accepting the last minute of invitation. Really, we have to hear from you a lot because you have vast research experience and contributions made in the field of regenerative medicine using different models as well as the experiments to design and prove the concept regenerative medicines from the earthworm to the human. Definitely, we are hearing from you a lot during the Global Leaders Conference and Award Ceremony going to held on 14th September. We are willing to hear as a research professional like you to encourage the audience and making the sense to learn the research and development, also a contribution made for sustainable development through innovative products or innovative materials or innovative concepts, which are very much important for the sustainable development goals to achieve. Thank you so much for your wonderful talk.
Yeah, thank you, sir. I love to join with you and support you in all the way. And your team is really doing fantastic. And so my support is there for always for you always. Thank you for the opportunity.
Thanks.
Thank you.
Thank you.
Yeah.